All right, here they come.
Okay.
All right. So for those filtering in so far, we're waiting on some other folks to filter in, and then we'll get started. So hang tight for a moment or so, and then we'll get going. All right. So let's go ahead and get started. So first off, I just wanted to thank everybody for joining and welcome to the webinar, which coincides with our launch last week of our AIM-HER2 algorithm via our AI site IMS platform. Title of this webinar is Introduction to PathAI, AIM-HER2 algorithm, and AI site IMS. Regarding questions, I know that if there's something pressing, feel free to drop them in the Q&A, but we'll also have time at the end for a Q&A session as well, a live Q&A session. So before I introduce our speakers, myself, I am Jeff Dunklin. I'm responsible for developing and supporting lab partnerships, as well as the deployment of PathAI's digital diagnostic solutions, including AI site, as well as our menu of algorithms across the Western US and select countries in Latin America and the EU. Been at PathAI for going on 7 months now, but started in diagnostics about 20 years ago in the specialty diagnostic genomic testing space. We had the opportunity to launch several different tests and technologies, including several single gene markers well before NGS, NIPT testing, and liquid biopsy, and now really excited to be here and really in the new frontier of digital pathology and AI. Our two speakers today, first will be Dr. Eric Walk. He serves as PathAI's Chief Medical Officer, where his strategic vision and leadership have been instrumental in navigating the complex and ever-evolving landscape of digital pathology and AI in healthcare. Dr. Walk oversees our entire medical team, including 20 pathologists at our AP lab in Memphis, PathAI Diagnostics. He's got over 20 years of experience in precision medicine, oncology drug development, and IVD companion diagnostics development. Dr. Walk is a board-certified anatomic and clinical pathologist, as well as a fellow of the College of American Pathologists. Currently, he's a member of the CAP Personalized Healthcare Committee, as well as the CAP Digital and Computational Pathology Committee. Dr. Walk's vast knowledge, experience, and dedication to improving patient outcomes through companion diagnostics, precision medicine, and now AI make him truly an insightful speaker. So thank you for being here, Dr. Walk. Second up will be Santosh Balasubramanian. For over 2 years now, he's served as PathAI's Director of Algorithm Products. He has degrees in computer science as well as engineering biology from Princeton University. Santosh leverages his diverse educational background and experience in computer science, machine learning, and AI to drive advancements in AI-powered pathology and precision medicine. Notably, his experiences as a cancer researcher at the City of Hope, as well as his experience as a bioinformatics researcher at Stanford University, where he designed and built the machine learning pipeline for Stanford's clinical decision support system. Santosh will share his perspectives, showcase some features within our IMS AI site, as well as do a thorough demo and showcase PathAI's AIM-HER2 algorithm outputs. So thank you both for being here, and Dr. Walk, take it away.
Thank you, Jeff, for the very kind invitation. I should have invited my mom to this webinar so she could have heard that. And welcome, everyone, and thank you for joining us for this webinar. We're really excited to see that we have over 100 people registered for the session today. So as Jeff mentioned, the goal today is to give you an overview both of PathAI, but more specifically on our recently launched AIM-HER2 breast cancer algorithm, AIM standing for AI measure of HER2. Before I get to the webinar proper, I do have a legal disclaimer slide here. You'll be excited to know I won't read this entire slide. Feel free to screenshot it and read it later. I will say that the information presented here today is for general purposes only and should not be construed as professional or medical advice from PathAI. And importantly, products and services discussed in this webinar are for research use only and are not intended for diagnosis or treatment. With that, here's our agenda for today. So as you can see, we have 5 topics. I will cover the first 3, being a quick overview of PathAI and our algorithm menu and strategy. Secondly, I will give a very brief 101 on machine learning methods, and then get to the heart of the webinar, an overview of the AIM-HER2 breast cancer algorithm. I'll then pass it to my colleague Santosh, who will review and provide an overview of our AI site image management solution, and also give a live demo of HER2.
Can you share your screen? The slides.
Oh, I thought I was sharing the screen.
No, not yet. There we go.
Try again. How's that? Can you see it now?
Yes. Thank you.
Okay, if that's the case, then I'll go back to this slide and say again that the information presented today is for general purposes only and should not be construed as professional or medical advice. And also the products and services discussed here are for research use only. So a brief overview of PathAI. And I will keep this brief. The company was founded in 2016 by Andy Beck, who is a pathologist with interest not only in pathology, but also in machine learning. And it was really his vision that put us on the path that we've been on for our entire existence, really focused around this mission and vision to improve patient outcomes with AI-powered pathology. And as a company, we're laser focused on this mission and have really focused on 3 areas, being unlocking the full potential of biomarkers from histopathology images, and we'll give an example of that today with HER2. Secondly, we're really passionate about bringing what we call precision pathology to end users in pathology labs and other pathology institutions. And by precision pathology, we mean not only a more precise version of pathology with regards to biomarkers, but also bringing characterization of cell and tissue features at scale to the masses. And by doing these things, we firmly believe that this will lead to improving diagnostic accuracy and, importantly, time to optimal treatment for patients, which, as you all know, is a bit of a gap that we have now, especially in certain diseases. Brief overview of the company from a financial perspective. We are a pre-IPO company. We raised over $350 million from various strategic and venture investors that you can see on the slide. From a product and services perspective, we have over 100 products and services across 3 categories. Our bread and butter, if you will, are robust, high-quality algorithm products, and we'll talk about HER2 today. We'll also touch on the platform where those algorithms live and exist, and that's called AI-Site. We have a version of AI-Site called AI-Site DX that's been 510(k) cleared and CE marked. And then the third category is lab services. So we own and operate the 3rd largest anatomic pathology independent laboratory in the country. That was called Popular Healthcare and is now called PathAI Diagnostics. And we're doing some really interesting things in this laboratory, which include, of course, implementing AI into a clinical workflow. But we've also developed a CAP-CLIA Pharma Services CRO laboratory, so we can deploy AI solutions directly into clinical trials. From an employee perspective, we have over 700 employees across 4 different divisions: translational research, clinical development services, diagnostics development services, and digital diagnostics distribution. And these 4 areas of the company really also nicely represent our philosophy and strategy for developing algorithms, starting with translational research. So we do quite a lot of translational research with academic experts, but also pharma companies. And we have a team of translational scientists that work with scientists in these other organizations to really vet different hypotheses that might suggest the use of an AI tool or a digital pathology tool in cancer biology or in the biology of other diseases. Some of these hypotheses then are proven out and lead to the opportunity to work on clinical trials in these areas. So this is why we acquired the laboratory and built a CAP-CLIA CRO laboratory there, so we can deploy early algorithms into phase 2 and 3 clinical trials. And some of these opportunities then show promise as IVDs or even companion diagnostics. So we put together clinical, regulatory, and quality infrastructure that allow us to develop algorithms under design control and bring them through regulators like the FDA to the market as an IVD application. And then last but certainly not least, we're focused on commercialization. So our commercial team is called Digital Diagnostics Distribution, and they are all about bringing our IMS and algorithms to end users. A lot on this slide, but it's really meant to highlight the ways in which PathAI as a company is differentiated from other solution providers in the marketplace. And it really comes down to 3 areas of data, annotations, and development. And we're really proud of the infrastructure that we put together to develop high quality, robust algorithms. And it really starts with the data. So I won't go into the details here, but you can see that we put in place a number of important partnerships with different organizations to get access to a large amount of slides, around 50 million at this point, some of which have important metadata associated with them, including molecular data or outcome data that are really important to developing novel algorithms. But as impressive as having 50 million slides is, they're really not very useful unless you can extract histopathology information or annotations from them. And that's where our contributor network comes into play. So at this point, I just got an update. We have over 550 pathologists who are contract pathologists for us. And these pathologists work directly with our machine learning teams to help annotate slides that really are the ingredients for our algorithms. And then on the development side, we're very proud to have one of the largest teams across the industry, comprised of software engineers and machine learning engineers who spend every single day thinking about the best ways to apply machine learning technologies to problems in histopathology, including inventing new machine learning methods, which actually is relevant to the HER2 application. This is our current menu, which is always in flux. Of course, the focus today is the HER2 breast cancer algorithm, but just a brief overview starting on the left. The oncology H&E area is very exciting. So the idea here is to extract novel information from H&E slides purely based on morphology, some of which really is not accessible by human eyes. So I've listed some Examples here, the tumor microenvironment. We just launched an application called PathExplore across 8 different tumor types that at scale characterizes the tumor microenvironment, tertiary lymphoid structures, and an exciting area called molecular predictors, where it may be possible to predict the likelihood of a molecular alteration based on an H&E. In the middle, oncology IHC, I think this is probably obvious that it's important to develop characterization and quantification algorithms for some of the common IHC markers. I won't go through these all, but you can see this is weighted to immuno-oncology and the breast panel. And then on the right, just wanted to touch on some of the non-oncology work that we're doing in non-alcoholic steatohepatitis, for example, including in clinical trials. Inflammatory bowel disease is an area that can benefit from AI, as well as other inflammatory diseases. Multiplex technology, I think, is an emerging area where AI can really have value in terms of really applying spatial techniques to multiplex IHC assays and really take it beyond simple quantification. And then I'll touch briefly on some of the workflow products that we're working on. I did want to, before I get to HER2 itself, really just wanted to share the vision that we have for AI pathology in general, starting with this workflow, which I'm sure you're all familiar with, which is the standard workflow in histopathology, which really hasn't changed over the past 50 years or even more. And what we're thinking of is a vision that really goes beyond simple linear digitization of all those cells in the workflow, but what are some new workflows that add value to pathologists, oncologists, and even patients that we can create with AI pathology. So there's a lot on the slide. I'm not going to go through it all, but I did want to touch on a few of these novel workflows because I think they're really interesting. Downstream of digitization, we're talking about applying AI algorithms of different types to those slides that create these new opportunities for bringing novel information to the pathologist even before they sign out the case. So things like reflex IHC ordering based on the tumor that's detected on the H&E level. Looking at NGS tumor sufficiency, this is a problem we hear from a lot of labs, NGS failures based on insufficient tumor. Can we help with AI? And then a different NGS application, screening from H&E, which is what I just referred to in the last slide as a molecular prediction. Intelligent case distribution and sorting is another area that we're interested to pursue. The idea that, especially in a large health network, we can sort cases even before the pathologist signs them out based on specialty or even difficulty. Some really innovative ideas here. What I'll focus on today is AI-assisted biomarker quantification of HER2, but this, of course, can apply to really any biomarker. And then a more tactical use case is QC and rescan. So to ensure the highest quality slides are getting to the pathologist, have an algorithm that looks for below quality slides, either at the histology level or at the digital level, and have those either recut or rescanned. So just to share with you some of the specific areas where PathAI is working on this, I'll touch on some of our applications. So we are working on an automated QC report for whole slide images, and this leverages our pre-existing artifact model, which is actually built into the HER2 algorithm. So this model detects usable tissue, background, blur, histology errors like tissue folds or crushed areas, and automatically will identify them and exclude them from the quantification. And impressively, this model was trained on over 500,000 annotations, 12,000 slides, 15 organs. And then I won't spend a lot of time on it because that's what I'll get into during the webinar. But we are really focused on IHC output and IHC quantification as part of the AI site platform. So we have a PD-L1 lung cancer algorithm deployed across 20 institutions in the US and the EU as part of our early access program. And we will be kicking off that early access program for the HER2 algorithm as well. So Little plug for that. So if anyone on the call is interested in that, please contact Jeff. So a quick 101 on machine learning methods, in case there are people on the call who are not familiar with how AI and ML methods are applied to histopathology. And I think this is really important for pathologists and any lab professional as we go deeper into the era of AI pathology. So just like you're all very familiar with the difference between IHC technology and PCR technology, NGS technology, you know the pros, you know the cons. I think it's equally important to become fluent in these AI ML methods. And certainly this is not a comprehensive overview, but really meant to be that first layer of the onion. So starting on the left are convolutional neural networks. And these are highly supervised methods. They're hypothesis driven, meaning you need to know what you're looking for, so you can train the algorithm to look for that as well. So PD-L1 is a great example of that, where we can have our contributor network circle PD-1 positive cells and PD-1 negative cells, and the algorithm over time will learn those things. The pro is that you have a high degree of explainability at the cellular level. The con is that this requires a lot of annotations, and you're not going to discover anything new with this approach because it's hypothesis driven. On the opposite end of the spectrum, you have end-to-end models, and one example of that is multiple instance learning, which is actually what was used for our HER2 model. This is a weakly supervised method, and it's hypothesis seeking. The advantage of this is you can, in contrast to the CNN, discover new associations that you were not aware of before. The limitation is that the explainability tends to be low, so it's more of a black box approach. And so applications that benefit from this approach are things like mutation prediction, and in this case, HER2, and I'll tell you the story of that. In the middle, you have an approach called graph neural networks, which is really in between because you're making some assumptions about items that are important, like cells and the distance between cells to create these graphs. And so really more of a middle ground. And then very quickly, generative methods are beginning to be used in histopathology. This is the underlying technology behind ChatGPT, and we can use this in histopathology. And I think we're just scratching the surface of how we could use this. I think what we're going to end up with is the ability to generate synthetic histopathology cases. But for now, we can use it for things like scanner image transform algorithms for development, for example. So very brief 101. And with that in mind, how do we think about biomarkers and AI pathology? And at least I think about it in two ways. One, we can use the techniques that I just showed you to enhance the scoring of existing biomarkers like HER2 or PD-L1. And I'll show you the HER2 example in a moment. But on the other side, we can do something that perhaps is even more exciting, and that's reveal and discover new biomarkers from histopathology images using approaches like end-to-end learning or graph neural networks. And I think there's a lot of potential here to really do some novel discovery of predictive, prognostic, and other biomarkers that were just not possible a decade ago. Okay, so now to get into HER2 itself, and I'll provide an overview of the algorithm, but I'll start with a little bit of background just in case you're not familiar with this. So this field has really been activated by this data, which was published in the New England Journal of Medicine. So this is the DESTINY-Breast04 phase 3 trial results of trastuzumab deruxtecan, otherwise known as AIM-HER2, which in this phase 3 registration study really showed activity that was very clear in metastatic breast cancer patients who were HER2 low. So from a progression-free survival perspective, there was about a doubling of the PFS, and then a very significant 6.5+ month overall survival benefit as well when you compared trastuzumab deruxtecan to physician's choice chemotherapy. So as a result of that data, the FDA approved AIM-HER2 for for HER2-low breast cancer in August, and a few months later in October, the FDA approved the Roche pathway anti-HER2 companion diagnostic for HER2-low breast cancer. Very recently, ASCO-CAP updated the HER2 guidelines on this 2023 update. I won't go through the details here, but at a high level, they affirmed the 2018 guidelines, but recommended a footnote be added to reports. And you can see that footnote here. They say it's premature to change the reporting technology for low levels of HER2 expression, but they do recommend a footnote indicating that patients with breast cancer that are HER2 low, IHC 1+ or 2+ish negative, that these patients may be eligible for a treatment that targets HER2 low. And to be complete and forward-looking, there are many HER2 ADC agents in clinical trials, and here's a reference and a table from that publication showing just how many there are in phase 1, 2, and 3 studies. And the difference between these agents tends to be variations in the antibody end, the FAB end of the monoclonal antibody, the linker, which can be of various types, and also, and importantly, the payload, which typically is a cytotoxic chemotherapy of different types. I don't have a slide, but I should also mention that these HER2 ADC agents are also being explored outside of breast cancer in different tumor types. So at the last ASCO meeting, the results or interim results from the pan-tumor02 study were presented for trastuzumab deruxtecan, showing promising activity across a number of tumor types outside of breast cancer. So to the algorithm. So as I mentioned earlier, our machine learning team initially investigated 2 ways to develop a HER2 algorithm. One that was convolutional neural network or cell-based approach focused, and another trying to develop a HER2 algorithm using a new form of multiple instance learning that our team developed called additive multiple instance learning. And so the bottom line is, when we did a head-to-head comparison of these 2 approaches, it was very clear that the additive multiple instance learning approach had better performance. So that is the technology that underlies the algorithm that you'll see in a second, where Santosh will do the demonstration. This additive multiple instance learning approach was developed in-house, and I won't go into the details here, but suffice it to say that it adds the advantage of a bit more explainability. As you remember, that is one of the downsides of typical multiple instance learning approaches. And this new additive approach brings some of that explainability back, which you'll see in the demo. Some specifications of the algorithm. So as you would expect, the inputs are HER2 IHC stained breast cancer specimens. These can be biopsies or resections, primary or metastatic. The IHC clones that were used in the training include 4B5 as well as Herceptest. Multiple scanners were used during the training. Importantly, from a regulatory perspective, this is an RUO algorithm, and the outputs are the HER2 score, 0, 1, 2, and 3+, also the area of invasive cancer, and you'll see the heat map overlays in the demo. Did want to touch on the training data. So an impressive amount of training data went into this algorithm, over 157,000 annotations from 12,000 scores, 4,000 slides, and a total of 65 plus pathologists were involved. On the right side, you can see that this algorithm, like all of our algorithms, are multi-layered. That artifact model that I described at the beginning is active here, automatically eliminating those areas that are folded, out of focus, blurred, etc. The tissue model is, as a pathologist, very important to me because this eliminates the need to have the human pathologist circle regions of interest because it's automatically identifying areas of tumor, stroma, etc. And I should add here that it's also been trained on in situ cancer in the breast, so it automatically will detect DCIS and LCIS and exclude those as well. And then, of course, you have the scoring model doing the HER2 prediction score, which you'll see. Quick shout out to our network of pathologist contributors. So they did a lot of work for this algorithm, over 12,000 slide scores, 4,000 total slides, 157,000 annotations. So we really appreciate their contributions. We have generated validation data for this algorithm that I'm showing here. So this was a comparison of the AI output of the HER2 score to manual scores by 52 board certified pathologists. There were over 700 cases involved, or slides, I should say, involved. All the pathologists had experience in HER2, and the analysis was done via a nested pairwise non-inferiority analysis, essentially a round robin comparison between AI and inter-pathologist concordance. And as you can see from the data on the right, the algorithm compares quite favorably to the average pathologist across the scoring bins for HER2. So with that, I'm going to turn it over to my colleague Santosh, who is going to first tell you about the workflow for AIM-HER2 breast cancer, and then go into a live demonstration of the algorithm itself. So Santosh, over to you. And I should add, Santosh is not feeling well today, so really appreciate him being a trooper and doing the demo. And so his voice is a little bit shaky, that's why. But thanks, Santosh. I hope you feel better.
Thanks, Eric. So first, talking through the workflow, we really designed this to sort of pair well with existing pathologist workflows, and not just for the pathologist, but lab technicians, for lab IT, and to make it as seamless as possible. So there's sort of 3 core steps to the process. The first is a lab tech will prepare the specimen, stain with HER2 as normal, and then scan the actual slide on their in-house scanner of choice. That WSI, or whole slide image, will then be uploaded to the cloud, where AISite will automatically ingest new images as they're being created. From there, the HER2 breast algorithm will automatically run and generate a HER2 score and the visual overlays that Eric talked through. And then the last step is the pathologist will have a queue of cases within AISite, and some of those may be HER2, some may be PD-L1, some may simply be manual reads if they're choosing to review some cases manually. And once they click into each of those cases, they'll have the ability to confirm slide adequacy to ensure that on-slide control tissue is being excluded from the analysis, and to review the actual algorithm score and overlays, and ultimately either agree with the AI provided results or disagree and provide their own score. And if they disagree and provide their own score, that sort of amended score is what will go into a report generated by AISite. And critically, that lab tech and process, those 2 columns, those are really designed to happen kind of in the background from a pathologist's perspective, such that they can happen overnight or in the morning, so that when a pathologist sits down, they're ready to just start going, and there's no other pre-work steps necessary before they start their analysis. Next slide. Briefly, a little bit on AISite. So this is our cloud-native image management solution that was really designed from the ground up for sort of an AI-based world, thinking about the fact that there are increasingly going to be algorithms used in histopathology, and we wanted to design an image management solution that sort of provides native integrations and keeps that AI front and center, and makes it easy to use, and as easy to use for the pathologist, but is also flexible enough to change the workflow depending on the specifics of the algorithm, because we know that the type of experience that you would want when you're scoring HER2 versus when you're scoring PD-L1 versus maybe when you're scoring ER, PR, Ki-67, like those might all be different. And so we designed AISite to provide the flexibility necessary to support a variety of algorithms. The particular version of AISite that you'll see today, as Eric mentioned, is our RUO platform. But we also have a regulated version of the IMS called AI Site DX, which has received FDA 510(k) clearance and CE mark in the European Union for primary diagnosis. Next slide. And I think from there, I'll transition just directly into the demo. So give me a second to share my screen. And then we can talk through some of the details of AI Site and AIM-HER2 breast cancer. So I just shared my screen. And so what you're looking at now is kind of a test environment for AI Site RUO. This is the pathologist's dashboard. So here you can see a number of accessions, which are provided with patient IDs, but also critically the tests that have been ordered. So you can see in this particular example, there have been some HER2 breast cancer tests ordered, PD-L1 in UC, PD-L1 in non-small cell lung cancer, melanoma, et cetera. And you can see the status for each of these cases. As I mentioned, the whole workflow is designed such that the pathologist can wait for the rather than just waiting for the algorithm to process, they can just come in when they are ready and directly tackle any of the cases that are sort of awaiting pathologist review. So let me go into 2 example cases here. So this first case is a HER2 breast cancer case. And this is sort of the initial view that a pathologist gets when they load the case. You'll see that there's this area to exclude regions of tissue. And this was really designed to make it very easy to ensure that the AI is appropriately ignoring any on-slide control tissue. So in the background, the algorithm has already identified these different regions of tissue. And as you can see, has already pretty cleanly separated the control tissue on the left from the patient tissue on the right. So as a pathologist, all I would have to do is confirm, yes, these are the control regions, submit it for slide scoring. And because all of these results have been pre-computed in the background, I'm immediately able to see what that primary result of the HER2 score is in the slide results review panel. Most pathologists give us the feedback that they don't want to see the algorithm results initially. So we actually hide these by default and allow the pathologist to turn them on when they're ready. So let me first turn on these overlays. And these are the additive MIL overlays that Eric had described. And you can think of these overlays as at a very high level, capturing the underlying staining pattern within each of these regions of tissue. And in particular, capturing the consistency with a HER2 0 versus 1+ versus 2+ versus 3+ staining pattern. And to make it a little bit easier to review these algorithm results, I'm going to go ahead and turn on the side-by-side view, which allows me to see the original IHC on the right and the overlays on the left. And just for sort of ease of visibility, I'm going to enter full screen so that I have sort of maximum real estate for that the slide itself. And now if I zoom in a little bit, you can see in each of these regions some of the HER2 staining. And if I open the legend, you can see that in teal, in this tealish green color, we're seeing the areas that are consistent with HER2 1+. This is sort of a faint partial membrane staining. And then in these areas here, a more weak to moderate complete staining around individual cells. And so at a very high level, when I load the case, I'm able to get a sense of where the sort of intratumoral heterogeneity may be. And if I open the slide results review panel, I'm actually able to see a quantification of the area of invasive carcinoma, the area and the quantification of the percentage of the invasive carcinoma that is consistent with each of these staining patterns. In this case, the AI has identified that the majority of the case is consistent with HER2 1+. But as a pathologist, I would have the ability to either accept or reject the score. Just to go into another example case, here's another example. If I come into AI site, and then turn on these overlays. Here you can see the overlays are predominantly identifying consistency with HER2 2+. And so if I, again, turn on the side-by-side view, you can see the regions of that HER2 2+ staining that are being identified by the algorithm. And again, if I come in here, I can either accept or reject the score. And if I choose to reject the score, I can say that perhaps the algorithm is inaccurate, provide my own score as sort of an explanation of what's happening, and perhaps say that the AI is overscoring certain tissue regions. And then submit. And then I can open a preview of that report, which will show, that the result was set manually by me at this particular time and what that score was. So that's a very quick overview of a couple of cases. Let me stop sharing my screen. And in the remaining 15 minutes or so that we have, maybe we can go to Q&A.
And Jeff, I believe we do have some questions in the chat, right?
We do. We've got a few questions here. First off, Jack was wondering why the performance is higher with MIL compared to cell-based CNN models in regard to HER2.
Yeah, so one of the advantages of additive MIL techniques is that they are able to better account often for sort of variability in slides across sort of different groups within a cohort. So for example, we know with HER2, there's going to be some amount of variability in the staining properties and the staining patterns, and even in the image properties across different scanners, across different labs, across different cohorts. And one of the advantages and one of the reasons that we've chosen to pursue an additive MIL-based approach here is that we've seen that it is better able to generalize to new types of slides and new cases that were not part of the training set. So in our internal evaluation, we actually performed a head-to-head comparison of our additive MIL-based techniques with the traditional CNN approaches, and found that on a held-out evaluation test set, that additive MIL model outperformed the CNN.
Next question. Have you taken specific steps to handle storage of large whole slide images?
Yes, we have taken specific steps. We have a large sort of platform engineering group that really thinks about issues of performance and reliability, and really works with the lab customers that we've been talking to ensure that we're being efficient with storage, and that we're able to keep those storage costs down to the extent possible.
Thank you. What are the expected timelines of 510 clearance and IVDR approval for the AIM-HER2 algorithm?
Yeah, at this point, we do not have plans to pursue a 510K or IVDR submission for AIM-HER2. That could change, especially if we have a pharma partner interested to work with us on that. I don't know, Santosh, do you have any other thoughts?
I think the only other thought that I would have is that the advantage of having an RUO product at this stage is just the rapid nature of the feedback that we're able to get from labs. So for example, this feature that I demonstrated of being able to automatically identify the on-slide control tissue and sort of ignore that from downstream analysis, that was actually feedback that we received earlier on from our PD-L1 non-small cell lung cancer algorithm, as that being a critical thing when we were actually going to use that product in sort of an RUO setting with labs. And that type of feedback and being able to respond to that feedback in an RUO setting is really helpful and valuable for us. And so I think that's part of why we're taking things sort of one step at a time, and focusing on making sure that we're collecting the feedback and iterating on not just the model accuracy, but also the pathologist workflow experience, and that we understand the nuances of what would and what wouldn't work in the diversity of labs across the world.
And as I mentioned earlier, as an organization, we have the infrastructure, the capability in clinical, regulatory, quality to develop products under design control and bring them through on a regulated path. We're just strategically deciding on when to do that and when to wait, as Santosh mentioned.
So you may have, Santosh, you may have already answered this, but the next question is related to control tissue. So does the model look at the control tissue and interpret that as a QC before starting?
That's a great question. It's actually a request that we've received from pathologists and labs. Like, they want us to be able to look at control tissue and be able to flag failures in either the positive control, if there's excessive background staining in a negative control, or if there's insufficient staining in a positive control, and be able to flag that to a pathologist. And that's not something that this version of the product does, but it's definitely something that we're looking into, again, per that sort of continuous improvement mindset.
So the next question, maybe Santosh, you can help me here. Do you have any institution that MIL provides more explainability than strongly supervised CNN? Seems counterintuitive.
Yeah, so I would say that there are trade-offs between the two from a pure explainability perspective. If you're at very high power, it's obviously very difficult to beat like single cell predictions and being able to see what every individual cell was predicted as. But one of the advantages of the additive MIL method is that even at low power, it gives you a very easy way to see how the different regions of tissue have been characterized by the AI, so that a pathologist can choose, I'm going to zoom in here at a high power and really study this, or I'm going to focus my analysis at somewhat a lower power. And in interviews with dozens of pathologists, we found that for HER2 specifically, the additive MIL method and that method of sort of characterizing that high-level staining pattern better matches their mental model and helps them sort of understand what's happening under the hood, and also come to potentially more confident decisions.
And Santosh, maybe I can ask a question on behalf of the audience to react to that last question. But can you talk a little bit more about the area calculation that we're reporting? Because I think that's a really interesting feature, but I also think it's important for users to understand what that is and what it isn't.
Yeah, I think it might be easier if I share my screen again. So let me go ahead and do that. So yeah, so here's the case that we were looking at before, where I had sort of projected the results, and you can see the area consistency with HER2 0, 1+, 2+, and 3+. And maybe this is potentially a simpler case, but one of the key caveats of this approach is that if I were to zoom into these regions that are being categorized as, being consistent with HER2 2+, I'm very confident that you would be able to find an individual cell or two that had absolutely zero staining. And so it's very important when you look at these area consistency measures, not to interpret them sort of apples to apples with the thresholds that are in the ASCO CAP guidelines. So for example, if greater than 10% of invasive tumor cells are strong, complete staining, that implies a 3+. There's not any explicit line of code in the AI that says if this area is greater than 10%, call it X, because the regions that the AI is looking at are not, rather than individual cells, sort of tissue regions and patches that include multiple cells, and that allows the AI to sort of learn over time these high-level staining patterns. So that's a key caveat that we talk about with pathologists when we first introduce the tool, to make sure that you understand what type of information is being conveyed with this measures, and therefore how you can use it versus how you may not be able to use it.
Great, thank you.
Next question is around the staining as well. Have you detected differences in staining between the two clones used, 4B5 versus Hercept test, especially at the 0 to 1+ bins?
I'm not sure that we've done sort of a particular deep dive into that 0 versus 1, those bins in particular, and I wouldn't want to speculate here on some of those differences. But we have definitely observed differences potentially in batches of cases across multiple labs. For example, maybe in terms of the frequency of cytoplasmic staining versus just membranous staining, or in the contrast between the hematoxylin and the DAB. And so again, to the earlier question of why additive MIL, one of the advantages of this approach is that it's better able to learn in general sort of these batch level considerations and potentially account for some of those differences across data from different labs or different institutions.
Next question. Is the platform able to collate the IHC score with FISH results entered from elsewhere to provide a HER2 result for the report? And are the reports customizable?
So at the moment, the report formats are customizable in the sense that we can change what the report looks like for a PDL1 case versus a HER2 case. And we have a fair amount of flexibility there, but they're not yet customizable in the sense that Lab A can't redesign the report to make it different from how Lab B would want it. That's something that we've heard some labs express interested in. It's just not something that is currently a capability of the platform. And in terms of being able to collate IHC score with FISH results, that's an interesting suggestion. One of the sort of things that we've taken to heart is that when we talk to labs, we recognize that AISite and PathAI is not the center of the universe. Many of these labs have their own pre-existing LIS, and in some cases an IMS, and other sort of lab infrastructure. And so when we talk to labs, typically their interests are in the reverse direction, wanting to take results from AISite and sort of pipe them into other pieces of lab IT or lab infrastructure, so that it can sort of more seamlessly fit with their existing workflows. So that's not a capability that we have today, but something that we can consider in the future.
Next question is around the training set, it seems like. Regarding the annotation, what percentage of your slides are at the tissue level? What percent are at the cellular level? And what percent are at the subcellular level?
So I think one of the distinctions that Eric drew between the CNN-based approach and the additive MIL approach is that the additive MIL approach does not actually require any individual per cell annotations. And with traditional sort of algorithm development techniques, that can actually be a major expense. You're talking about tens, potentially hundreds of thousands of individual cell annotations. So for this particular HER2 breast model, there are definitely tissue level annotations, in particular to allow us to learn how to distinguish between DCIS, LCIS, invasive carcinoma, necrotic tissue, normal tissue, et cetera. And then use those as inputs into that scoring model. But there are not any annotations for at the cellular level or subcellular level.
All right. What if the model predicts equal percentages for 2+ and 3+ areas, for example?
It's a great question. I'm not sure that we've actually seen a case that has like that exact sort of decision boundary issue. But I think it's a great example of why we think it's important to be transparent about that and to show that to a pathologist so that they can maybe perhaps spend a little bit more time adjudicating sort of difficult boundary scenarios like that. In general, the model has ways of sort of learning directly from consensus slide level scores, sort of how to balance the considerations of what's 2+ versus 3+. And the advantage of having those HER2 density heat map overlays is that you're able to see exactly sort of what are the signals that the AI is using to come up with a final slide level score. All
right, questions, a few more coming in. Were measures taken to ensure that the training data were representative of the broad breast cancer population, patient population, both for clinical and demographic characteristics.
So we definitely took measures to ensure that we were getting as broad a training set as possible. And so like one of the examples of things that we did was make sure that we were sourcing data from external institutions. The other thing, or sorry, from multiple institutions. And then the second thing that we did was rather than just splitting our data such that, you know, we have some data from lab A, we train on lab A, and then we evaluate performance on more held out data from lab A. We actually held entire data sets out from the evaluation. So if we had, you know, 4 or 5 different data sources that went into training of the AI, we actually held out 1 or 2 of those so that we could test what we refer to as generalizability, the ability to have the AI perform well on data that was unseen. And we have, in some cases, clinical and demographic characteristics that we were able to use in sort of balancing the cohorts, but not across the entire data set. So to the extent possible, we've taken those measures, but there's definitely more evaluation that we would want to do to ensure that there aren't any sort of implicit biases in terms of particular clinical or demographic covariates.
I know we've got a number of questions still here, but in regard to time, I wanted to pull out a few of these. Can this AIM-HER2 be expanded to HER2 ultra-low cutoffs, which is used in the ongoing DB06 study?
Yeah, that's a great question. It was one of the things that we were thinking about as we were developing the product, and it absolutely can be expanded to account for that ultra-low. It would require a little bit of additional development work. It wouldn't sort of come right off the shelf, but that's something that as we continue to sort of see the interim and ultimately the final results from DB06 is something that we'll be weighing with customers.
And what is your experience with DCIS cases?
So as Eric mentioned, we collect sort of individual annotations of the tissue so that the AI is able to identify DCIS. And we have sort of built-in sort of cutoffs such that if there's not a sufficient quantity of invasive carcinoma, the AI actually provides a message back to the pathologist saying that there was an insufficient invasive carcinoma to score the case.
Awesome. So there's a few other questions here more around the more machine learning techniques, things like that. And I can follow up on some of those individually with folks that have brought those in. Eric, do you have anything to add before we wrap up?
Maybe I'll call out one last question, which is from Marilena. You mentioned that validation was done using specific antibodies and scanners. Have you seen any variability between them? What about the other antibodies or scanners? Will the algorithm give the same score?
So we've specifically done that sort of stratified analysis and have not seen any significant deviations in terms of performance either across scanners or across clones. But obviously, based on the specifications table that Eric showed, there are scanners that we haven't been able to evaluate on yet, and clones that we haven't been able to evaluate on. And so that's something that we're actively interested in. If partners have specific data sets that come from either scanners or clones that are outside of the current specifications of the product. And again, given this is an RUO product, we're very open to sort of doing pilot studies to evaluate performance and to see what would need to change to improve performance if there are cases where the performance is suboptimal.
Great. Well, thanks, Santosh. So we're at time, so I think we should wrap up the webinar, but thank you all for joining us here today. Really appreciate the high level of engagement and the fantastic questions. Jeff, do you want to let the audience know about any follow-ups? Will we make the recording available? How does that work?
Yep. So after this, I will send out the recording. It'll probably take 5 days or so. So expect a recording of the webinar early next week, and feel free to reach out to me if there's other questions or collaborations or conversations you'd like to have around not only PathAI, but also obviously our HER2 algorithm. And just a thank you to you, Dr. Walk and Santosh. Thank you both for being here.
Yeah, our pleasure. Thank you, Santosh, because I know you're not feeling well. So really appreciate you being here today. Thank you all for attending and hope to see you at the next webinar.
Thanks, everyone.
Take care. Thank you.
Bye-bye.